Hello, this is Article 2 presentation. Although DNA replication is a well-studied process, many aspects of its regulation remain unclear. One key player in replication initiation is CDC6, a protein essential for loading MCM27 onto DNA at origins of replication. Proper regulation of CDC6 is crucial for ensuring DNA replication occurs only once per cell cycle. In this study, researchers used biochemical purification and electron microscopy techniques to examine the properties of CDC6 and its interactions with other proteins. This research is important because errors in DNA replication can lead to mutations and diseases like cancer. By understanding the structural properties of CDC6 and its interactions with other replication proteins, scientists can identify potential therapeutic targets for conditions caused by these replication defects. Prior research on DNA replication has been limited due to X-ray crystallography, which does not provide the quality that electron microscopy, particularly single particle EM, provides. Our hypothesis is MCM27 is loaded onto DNA as a head-to-head double hexamer in a conserved reaction, forming topologically linked complex that can slide along double-stranded DNA passively. This is done through the help of ORC, CDC, and CDT1.
Our first method is protein purification. To investigate how MCM27 loads onto DNA, key proteins including ORC, CDC6, CDT1, and MCM27 were purified. ORC was isolated using tandem affinity purification, while flag affinity purification was used for MCM27. Once purified, the proteins were verified using SDS-Page and Western blot analysis. The purification workflow is shown in Figure S1. Ensuring high purity proteins was critical for accurately reconstituting the loading mechanism in vitro. The second method we identified was the reconstitution of MCM27 loading. The helicase loading process was reconstituted in vitro by incubating DNA templates containing replication origins with purified ORC, CDC6, CDT1, and MCM27 in the presence of ATP. DNA templates were generated using either restriction digestion for linear DNA or PCR amplification for circular templates. This experiment was designed to replicate the physiological conditions under which MCM27 loading occurs. Figure S4 illustrates the different DNA templates used in this study. The third method was electron microscopy analysis. Negative stain transmission electron microscopy was used to visualize MCM27 loading. Samples were stained with uranyl acetate and imaged at high resolution. The acquired images were processed through 2D classification and multivariate statistical analysis, allowing the distinction of structural details of the MCM27 complexes. 3D reconstructions were generated to analyze the hexameric arrangement. Figures S2 and S4 display the EM classification and structural arrangement of the helicase complexes. Our final method was ATP dependency assays and mutant analysis. ATP hydrolysis is thought to play a crucial role in helicase loading. ATPase activity assays were performed using thin layer chromatography and colorimetric methods. Additionally, mutant MCM27 complexes with defective ATP binding sites were analyzed to assess their role in loading efficiency. These results indicated the ATP binding and hydrolysis are essential for helicase loading. Figure S2 highlights these key findings.
We determined that the most important experiment of the paper was the recreation of the DNA loading process in vitro with various stipulations and tests. This would not only help indicate what was necessary to load the MCM27, but also give detail into which order each component was used in. So Figure 2 just goes over kind of the general requirements for the MCM27 loading. Figure 2E, which used silver-stained SDS-Page analysis of the loading reactions, demonstrated that the in vitro replication was efficient enough, and it validates the rest of the results for the experiment. Panel A is also important as it shows that the stable loading of the complex is reliant on ATP, with all of the required components for the loading process, those being the ORC, the CDC6, and the CDT1. And of course, the MCM27. The binding was done either in the presence of the ATP or in the presence of ATP gamma S, which is a analog of ATP, but is not hydrolyzable. And this showed that in the presence of ATP, the MCM27 was still bound even after a high salt wash. And with the ATP gamma, the MCM27 was not bound. So it emphasized that the ATP was needed for stable binding to the DNA. These findings are also emphasized in the experiments in panel B, which shows that MCM27 is loaded as a complex instead of each individual part. Because in the presence of the ATP gamma S with the high salt wash, all of the MCM components were removed. This was found through Western blot analysis and the high salt wash, as I mentioned. Figure 2C is also important as it shows that the complex binds directly to the DNA, as it remained bound even after the Echo R1 digestion. If it was not bound directly to the DNA, like it would have just fallen off, but even in the presence of that, it remained bound. A lot of the key details of the order of the binding were found in figure 3. Figures A and B showed that the CD6 and ORC were both integral to the MCM27 loading by testing the percentage bound at increasing concentrations of both proteins. That was also done in the presence of ATP and the ATP gamma S, as well as with the different salt washes. B is also important as it shows that the ORC is needed for the CDC6 to bind. And that gives an insight into the order and tells us that the ORC actually binds first and then the CD6 loads. And again, both are needed for the MCM to bind. One of the most interesting insights into the loading process is actually found in figure 3D, which discusses the sequence specificity needed for the ORC and inherently the MCM27 to bind. The loading process was recreated with the wild type DNA that was used in all of the other experiments versus a mutant DNA that had a mutated origin. These results show that the ORC and the MCM27 can actually bind both types of DNA equally in the absence of a competitor, which is this section of the table right here. That here we can see the ORC at varying concentrations, and it shows that the MCM27 still binds to the DNA. These findings, again, were especially interesting as they prove that the specific sequence of the binding site was actually not necessary for DNA replication in these cells, which allows for replication to occur even in mutant DNA with the mutated origin. However, when a competitor DNA was introduced with the mutated DNA, which you can see in this part of the table here, the competitor DNA was the wild type DNA, and it shows that the ORC and the MCM27 preferentially bound to the DNA with the wild type origin and not the mutated DNA, as you can see by the absence of that here. And it shows that in a physiological context, even though the ORC can bind to a nonspecific DNA sequence, it does prefer to bind to its native binding sites. Overall, the replication of the MCM27 loading process in vitro provided a lot of insight into the proteins and chemicals necessary for proper and stable loading, as well as giving an idea of the order in which these proteins are used and where the replication process can even begin in the DNA. These experiments provided like a detailed biochemical framework for the process of the replication origin licensing. and allowed for further analysis into how the MCM27 was loaded and its relationship with the DNA itself, which I believe will be discussed in the results.
Figure 4C on the left and supplemental figure S2 on the right both indicate that MCM237 is loaded onto DNA as a head-to-head double hexamer when the origin replication complex and protein CDC6 are present. In electron microscopic imaging, the supplemental figure shows when all the necessary components are present, DNA, ORC, ATP, and CDC6, there is proper formation of double hexamers, which are enclosed in the circle. However, without DNA, there is no formation, suggesting that DNA is necessary for the assembly of the complex. And if a non-hydrolyzable ATP analog, ATP gamma S, is used to replace ATP, there is also no formation, suggesting that ATP hydrolysis is necessary for proper MCM237 loading as it drives conformational changes in one direction. In the absence of ORC, there is also no double hexamer formation, which confirms ORC is required to recruit and load MCM237 onto DNA. Lastly, in the absence of CDC6, there is also no observation of MCM237 double hexamers, which illustrates that CDC6 is necessary for the assembly of pre-replicative complexes. In all, this indicates that the loading of MCM237 is a coordinated process that requires all components, and that ORC and CDC6 must work together to recruit and load MCM237. Figure 4C more closely depicts the double hexamer complex and its head-to-head formation. After reconstituting loaded MCM237 complexes with purified yeast proteins in vitro, a high salt wash was used to remove unbound proteins and only leave stably loaded MCM237 on DNA. Once the DNA was enzymatically cleaved using restriction enzyme ELCO-R1 to release the complexes for imaging, the MCM237 particles were negatively stained and imaged by electron microscopy. The images were used to generate class averages, which is essentially an average of multiple similar particle images to get a stronger signal clarity, containing about 100 particles. Averages in the bottom row were obtained from particles done in whole leading reactions, and high salt wash step was omitted to test if the double hexamer was the result of hydrophobic interactions due to exposure to high salt or simply due to the mechanism of the loading process. The reactions were incubated with 1 kilobase DNA fragments and then analyzed by electron microscopy. Both images are structurally indistinguishable from one another. From the side, the particles appear to be composed of 4 layers, with the 2 outer layers thicker than the 2 inner. The protein density along the long axis through the center of the particles was reduced, suggesting that there is a channel that runs through. The height of this MCM237 complex is twice that of the 2-layer CDT1 MCM237 complex, which suggests that MC237 is a double hexamer. The thinner arrows mark the inner and terminal tiers, while the thicker arrows mark the thicker outer tiers containing the AAA plus domains. This suggests that MCM237 are loaded head-to-head in a concerted manner, as the class averages show that MCM237 does not exist as single hexamers after loading, as there are no intermediate states. The channel present through MCM237 was further investigated, and Figure 6D provides experimental evidence that MCM237 is topologically linked to DNA and can slide passively along the ends of DNA. This is done by loading MCM237 double hexamer proteins onto either linear or circular DNA in vitro and checking their tension periodically over an hour in a buffer that mimicked physiological pH in conditions. The closed triangles indicate circular DNA with no ATP, open triangles are singular DNA with ATP, closed squares are linear DNA with no ATP, and open squares are linear DNA with ATP. The half-life of MCM237 double hexamers on linear DNA was approximately 35 minutes, which is partially due to electrostatic interactions between it and DNA, but it was still significantly shorter than the half-life of MCM237 double hexamers on the circular DNA, which is greater than 60 minutes, suggesting that it can slide off the ends of linear DNA and thus engages with DNA topographically. There is also minimal difference between the ATP and ATP-free reactions, which suggests loaded MCM237 can slide freely in an ATP-independent manner. This all combines to suggest that MCM237 may act during replication as a double-strand DNA translocase, translocating DNA from AAA plus domains toward the N-terminal domains in each hexamer, or even could allow for storing an active helicase that is stalled at a fork, critical for DNA replication.
Our next unresolved question is, how is helicase activation regulated post-loading? Future studies should focus on identifying molecular triggers that initiate replication and how additional factors contribute to helicase unwinding activity.